Biology 1108, presentation number 11, Plant Structure and Growth. In this particular presentation, we're going to be looking at specifically land plants, members of the kingdom Plantae, the tracheophytes. Remember, those are plants that have xylem, phloem, and a cuticle. And these are the far majority of plants that you and I are familiar with. So let's take a look at the structure of the plant body. The plant body consists of an above ground portion called the shoot system and then a below ground portion called the root system. We are going to be focusing at the present on the above ground portions of the plant, which consists of stems, branches, and leaves and things. The shoot system consists of those structures and are all generated by growth of what's called a terminal bud or an apical meristem. We'll be talking about this more at a later time in this presentation. The terminal bud or the apical meristem, apical means a tip, is a little region of growth found at the end of any given stem. So if you take a look at our tomato plant there, and our tomato plant consists of one single stem that seems to be growing upwards. At the very tippy top where it says shoot tip there, that's where I would find my terminal bud or my apical meristem. It is a zone of cell division that will ultimately give rise to all of the other cells and all of the other tissues that I see below it. So if I take a look at that shoot system, I notice it is divided into what we call nodes and internodes. Now, nodes are places where the action is. And I know that's kind of a trite way of saying it, but if there's a leaf, it's a node. If there's a flower, it's a node. If there's a branch there, it's a node. If there's an auxiliary bud, in other words, it's a bud that will create a lateral branch, it is a node. Nodes are where the action is. If there's a leaf, a fruit, a flower, a branch, it is a node. Now, in between the nodes are stretches of the stem that are called internodes. Notoriously boring because there's nothing going on. So the shoot system consists of the above ground portions of the plant, largely composed of stems that are ultimately generated by cell divisions in a terminal bud consisting of nodes and internodes. And the purpose ultimately of that shoot system is to bear and position your photocollectors, which are your leaves, and so the leaves are also a very functional, important part of the shoot system. Below the ground, of course, there's the root system. We'll talk more about roots in a little bit. But right now, make sure you are aware of the components of the above ground portions of the plant body. And if this particular illustration is not agreeable to you, then Google for another because there are hundreds of them out there. What we will be doing on the next 3 slides is take a look at the 2 major parts of the shoot system, the stems and leaves, and talk a little bit more about their functions, and then we'll talk about the roots. So we'll be taking a look at stems, leaves, and roots on the next 3 slides. Stems, you remember, are part of the shoot system, and their job is to largely support the leaves because the leaves are the photocollectors for the plant. But the stems are also going to be an agency of transport because obviously I am transporting water from the roots to the leaves, and with that water I'm going to be transporting nutrients as well. But then again, I'm going to be producing food in the leaves and transporting that downwards to other regions of the plant. So next to the bullet point transport, you might want to scribble down the words xylem and phloem. And remember, not too far ago, we talked about tracheophytes, which are plants that have vascular tissue. So the vascular tissue will be located in those stems, and so therefore our stem is an agent for transport. Stems can also be an agent for storage. If you take a look at the cactus plants down there, their stems are modified for photosynthesis, but they are also going to be modified for storing water. And you can actually store starch and food molecules and a variety of other things in stems as well. And as I had mentioned before, those cactus plant stems can be functioning photosynthetically. As a rule, if the stem is green, it's going to be doing photosynthesis. In the case of the cactus plants, they really need the stems to be photosynthetic because you've opted out of the photosynthesis role for the leaves because your leaves are now modified into those protective, technically non-photosynthetic spines. And then we've got protection. Now, if you take a look at the bottom left, you will see what amounts to being a thorn. Technically, thorns are modified stems, and they will contain vascular tissue. Now, another structure you're familiar with that's commonly called a thorn, like the thorns of roses, are actually not thorns. Roses don't have thorns because those sharp things on roses do not contain vascular tissues. The structures on roses are officially called prickles. And I know that doesn't sound terribly romantic, but in terms of roses, the things that you and I would commonly call thorns are not thorns because they're not modified stems. They're actually epidermal outgrowths. And so roses have prickles, not thorns. The plants that have thorns, when you look at them, the thorn will actually appear as a single stem-like structure or a branched stem-like structure that will have some pointy tips to it. It's like that honey locust, I believe, is what it is on the lower left. Leaves, part of the shoot system as well, function in photosynthesis, but also can function in storage. For example, looking at the jade plant at lower right, you've got some fleshy leaves that could be storing water. And then finally, for protection, in looking at the cactus plant, spines are modified leaves. Now a little bit more about leaves. Leaves have a stalk, and that stalk is called a petiole. And the actual photosynthetic portion of the leaf is called a blade. Sometimes it's called a lamina, Lamina. But in most leaves, the blade is a single structure, but in others, it might be multiple structures. So you will see below the two bullets, simple leaves and compound leaves. A simple leaf is a leaf in which the blade is a single laminar structure, like a maple leaf or like an oak leaf. And then we've got compound leaves. Now, in a compound leaf, the blade is composed of a number of different parts. And those parts, called leaflets, can be joined at one common location. In other words, a palmately compound leaf is a leaf where the leaflets, or the divided portions of the leaf blade, are all connected at one place, much like your fingers are all connected to the palm of your hand. And then there is a pinnately compound leaf, in which case the leaflets are attached either oppositely or alternately along a midrib or a central axis called the rachis. For example, hickory trees, or a green ash, or a walnut tree. So I see that illustrated to the right. Roots, as part of the below ground portions of the plant, are going to have a number of important functions related to life on land. In first case, life on land means you're dealing with gravity, which means the plant has to be maintained in a certain position relative to the sky and the sun and all that. So the roots are going to be functioning as a means of anchoring the plant in place. Anchorage is an important component of root function as well as absorption, because the root's going to need to be absorbing water and nutrients, minerals specifically, dissolved in that water, which are going to be transported upward to the plant to provide for the photosynthesis and also the life and growth of the above ground portions of the plant. And finally, storage. Roots are a really, really good place where the plant can actually store the products of photosynthesis when it's not doing photosynthesis. So in case we have a transition to fall and winter is just around the corner, photosynthesis is decreasing may have stopped, and so we're going to need to have some kind of supply of stored sugar, that is starch, in order to keep the plant alive during the fall and the winter. And in most cases, the excess sugars produced during the spring and the summer are going to be stored in the below ground portions of the plants. There's usually a significant amount of starch stored in root tissue. And so during the fall and the winter, that starch is going to be broken down into sugars, and those sugars will be transported through the phloem upwards to the cells that are going to need that supply of food. Now, a little bit more about roots. And you take a look at a root system of a plant. Root systems come in two different types. There are taproot systems and there are fibrous root systems. Taproot systems are more characteristic of dicots. You remember we distinguished between dicots and monocots not too long ago. A taproot system is a root system where you'll have 1, 2, or 3 major roots and then smaller root branches coming off of those. So you got a hierarchy of roots in terms of age and size. On the other hand, a fibrous root system, more characteristic of the monocots, is a situation where all the roots have the same basic diameter. And so what you see on the right is a tangled mess of roots versus what you see on the left, which seems to be a root system composed of definitely larger and smaller roots in some degree of order. Now finally, some cases roots can form where roots ordinarily aren't meant to form. So let's take a look at the illustration. I see a plant growing along the ground. Could be a vine, could be a plant that's fallen over. And in many cases, if a plant does fall over or a vine is growing along the ground, you will have the formation of roots directly from the stem. And those roots are commonly referred to as adventitious roots. An adventitious root system is a root system produced by some place in the plant where roots are not ordinarily found. So for example, if you were to take a look at English ivy growing up a brick wall, English ivy, take a close look at that. All of those stems are covered with teeny tiny little roots that essentially grow into the wall, brick, rock, and hold the plant up. Same thing could be true for like poison ivy. You ever seen a poison ivy vine growing up of a tree in a forest? And poison ivy vines are usually these big, fat, furry looking things. Well, you look closely at it and don't get too close, but the furry stuff on the stems growing up the tree in the forest are adventitious roots. And at this point, the poison ivy is getting its water from the surrounding atmosphere. So roots can basically pick up moisture from the air around them. And adventitious roots are notoriously famous for that. Now, regardless of whether we consider plants having a shoot system consisting of leaves and stems or a root system, plants have 3 basic tissue systems, and these 3 tissue systems are found in all of these plant parts, the above ground portions and the below ground portions. And we'll take a look at these on the next series of slides. Plants have 3 tissue systems. They are the dermal system, the vascular system, and the ground tissue system. The dermal system, as the name implies, involves the skin of the plant. Now, I use that in quotes because it's really not skin as you and I have skin, but it corresponds to the tissue covering the surfaces of the plant. And this would include epidermal cells, and epidermal cells as a rule are cuboidal in shape, and they're pretty nondescriptive, but they do secrete the cuticle. And you'll remember the cuticle is a waxy layer that seals the plant tissues against desiccation. It's one of those adaptations to life on land. Then sometimes these epidermal cells will produce hairs, and those hairs are called trichomes. And sometimes they're long and soft, or sometimes they're short, sometimes they're bristly. Sometimes their job is to prevent herbivory or animals eating up on the plant. Sometimes their job is to cut down the amount of ultraviolet radiation striking the plant. There are root hairs associated with epidermal cells in the roots, and these, like trichomes, are long finger-like projections growing from the epidermal cell. But the job of the root hair is to increase absorptive surface area. And so root hair can tremendously increase the amount of absorptive surface area for the individual epidermal cell, and therefore tremendously increase the potential for absorbing water and the potential for absorbing nutrients and minerals. And then finally we have our fourth bullet, which are guard cells. Guard cells are modified epidermal cells, and their job is to regulate the movement of gases into and out of the leaf. As a rule, guard cells are found associated with the epidermis found on the lower leaf surface. Generally don't find guard cells anywhere else. And the guard cells actually define openings, and those openings are called stomates. Stomates are like little windows, and what they're going to do is they're going to allow oxygen in, carbon dioxide in, oxygen out, carbon dioxide out. And the other thing is a stomate can be an opening through which water vapor might be lost from the plant as well. As a consequence, the stomate is going to need to be regulated, particularly on very hot, dry days. If you can close the stomate, you reduce the amount of water loss from the plant. And this is the job of the guard cells. Guard cells, as you see them indicated in the illustration, are allowing gases in and out, but we got to understand there is the potential for loss of water vapor as well. But those guard cells are regulated osmotically, which means I can move water into a guard cell, increase its turgor pressure, or I can move water out of a guard cell and decrease its turgor pressure. If I increase a guard cell's turgor pressure, the guard cells, which are essentially almost kidney bean shaped, will blow up. They'll full of water, and they will open the stomate. So increase in turgor pressure, guard cell fills up, stomate opens. And then in cases where we might want to reduce the potential for water loss, we can basically decrease turgor pressure in the guard cell. The guard cell becomes flaccid. The guard cells collapse upon one another, and the stomates close. So the dermal tissue system consists of those cells that actually form the outer covering of the plant. The far majority of those are epidermal cells, but we have these specialized cells, trichomes, root hairs, and guard cells that all qualify as modified epidermal cells, all part of the dermal tissue system. Now we're going to spend a bit of time on the vascular tissue system because it's pretty darn important. Vascular tissue, you'll remember, consists of xylem and phloem. Xylem's job is to conduct water and the minerals and nutrients dissolved in that water, largely obtained by absorption through the roots from water located in the soil. And then you've got phloem, and phloem's job is to conduct sugars, photosynthate, food molecules, generally dissolved in a solution of water, and the sugar solution transported through phloem is called sap. And the direction of xylem transport is always upwards, whereas the direction of phloem transport is potentially bidirectional. Sometimes it's transported upwards, sometimes it's transported downwards. And we're going to see in a bit that has a lot to do with what's going on, what time of day it is, and what time of year it is. So on the next couple of slides, we are going to be focusing on what is xylem and what is phloem. Xylem cells come in two different types. There are tracheids, which are long and narrow, generally with kind of pointed, slanted ends. And then there are larger diameter xylem cells called vessel elements that are usually a lot more truncated at the ends. So you will see to the right an illustration of tracheids far right and vessel elements a little bit to the left of those. Now, the thing is tracheids are considered to be the primitive cell type in xylem. The reasoning is if I take a look at ferns, and you know that ferns are the most primitive vascular plants we got. If you take a look at ferns, their xylem tissue is exclusively made up of tracheids. The same thing is true for conifers. If you take a look at a pine tree, and you know that pine trees, while they are seed plants, are still more primitive compared to the flowering plants. If you take a look at the xylem tissue in a pine tree, likewise, you will see exclusively tracheids. Vessel elements are considered to be the derived form of xylem cells, and you will find vessel elements in the angiosperms only. So if you take a look at this scanning electron micrograph photograph of xylem tissue, you will see tracheids, which are the teeny tiny little holes, and vessel elements, which are the much larger holes. That is actually probably a section through something like an oak tree. So tracheids, the primitive cell type; vessel elements, the derived cell type. Tracheids, narrow diameter; vessel elements, large diameter. In either event, tracheids and vessel elements will have perforated end walls because they're essentially going to be lined up end to end. They're like little pipes. And so what I'm going to be doing is I'm going to be ducting water and the minerals and nutrients dissolved in that water upwards. And they're going to be traveling through these hollow cells. Now, if you're a hollow cell, technically you're probably going to be dead at maturity. There is no reason for you to have a protoplast. There's no reason for you to have cytoplasm, because you are essentially nothing more than a section of pipe. And that's what xylem cells are. They're sections of pipe. They are dead at maturity. And as a rule, they are also secondarily thickened with what we're going to call secondary cell walls. A secondary cell wall is an additional cell wall laid down inside of the primary cell wall. Generally makes the interior of the cell a little more narrow, but what it also does is it imparts a tremendous amount of physical strength to the cell. And so when you think about it, you got a pine tree or an oak tree, and the thing is hundreds of feet tall. There's a reason why that tree of that size is able to maintain its vertical position. It's largely because those cells in the xylem aren't going to bend, because they're all reinforced with this internal secondary cell wall. Now, looking at the little inset photograph at the bottom left, I see that xylem cells in that picture, it's a light micrograph. It's taken with a light microscope. You'll see that the xylem cells indicated by the red arrow are fairly big and hollow looking, and as a rule, xylem cells will be found towards the center of the stem. This is something that will become an issue a little bit later. So right now, we understand that xylem consists of tracheids or vessel elements. We find them both in angiosperms. Tracheids are going to be found exclusively in the pines and exclusively in the ferns, because tracheids are considered to be the primitive cell type. And these cells function in the transport of water and those things that are dissolved in that water. Ground tissue is our third tissue system, and it's pretty darn easy, because if it's not dermal and if it's not vascular, it's going to be ground. Ground tissue is everything else. There's a wide diversity of cells that qualify as ground tissue. The far majority of those cells are living at maturity, and they're just referred to as parenchyma cells. Parenchyma cells could be, for example, mesophyll cells in the middle of the leaf, whose job it is to do photosynthesis. Parenchyma cells could be storage cells in the stems and roots where we might store starch. As a rule, ground tissue is anything that's not xylem, phloem, or dermal tissue. All other cells. So if I take a look at the leaf cross-section at right, or I take a look at the root cross-section below right, or I take a look at the stem cross-section at the bottom, the far majority of cells in each of those photographs, and each of those plant parts are ground tissue cells. Ground tissue system is our third tissue system found in plants, and it's a pretty easy one to get a handle on because if it's not dermal and if it's not vascular, it's going to be ground tissue. So the far majority of cells in the plant are ground tissue cells. Now, as a rule, those cells are commonly called parenchyma cells. It's just nothing more than a technical name for living cells in a plant that don't belong to the dermal system, that don't belong to the vascular system, but engage in important activities for the plant. Let's take a look at two examples here. Parenchyma cells would include mesophyll cells. Now, to the right of mesophyll cells, you will see a cross-sectional view of a leaf. Now, that leaf has got an upper epidermis, dermal tissue. It's got a lower epidermis, dermal tissue. But all the cells in between those two layers of epidermis are called mesophyll cells. Literally means middle of the leaf. And those cells are responsible for doing photosynthesis. Mesophyll cells do photosynthesis. Mesophyll cells are parenchyma cells. These are part of the ground tissue system. If I take a look at the picture immediately below that leaf, it's a cross-section through a root. And the far majority of that root is storage tissue. If you look at that little narrow round circle in the middle of that root, that's where the vascular tissue is. All the other cells are storage cells. All the other cells are parenchyma cells. All the other cells are part of the ground tissue system. Let's take a look at that stem cross-section that we've seen on the previous two slides. It's the picture immediately below the bullet point that says all of the cells. Now you'll see those vascular bundles that we saw before. You'll see the xylem tissue that we had a red arrow pointing to. You'll see the phloem tissue that we had the same red arrow pointing to. But then you got all these other cells in between the vascular bundles and in the middle of the stem. That's all parenchyma. That's all ground tissue. So if it ain't dermis, and if it ain't xylem, it ain't phloem, all other cells are ground tissue cells. In our talking about these three tissue systems, we've mentioned a number of different cell types. And so where do these cells come from? Well, we kind of alluded to this not too long ago. I said at the tip of any stem, there is this localized zone of cell division. We called it an apical meristem, something located at the tip. So plants have meristems. Meristems are localized zones of cell division. That's a big difference between plants and animals. In animals, cell division is kind of diffuse. It's found all throughout the animal. We don't grow at the tips of our fingers or the tips of our toes. Our cell divisions occur everywhere. Now, in a plant, cell divisions occur at the tips of the stems or the tips of the roots. And so the plant grows taller and the plant grows deeper. We'll also see that in some cases, plants will have some internal meristems that will promote the ability of the plant to increase in diameter. So meristems are localized zones of cell division. They're localized zones of mitosis. Where are they located? They become located at the tips of roots, in which case the root grows deeper, or shoots. The plant grows taller. Or they can be located inside the plant body, in which they would increase diameter. So we're going to note this in a little bit, but you might want to write down the word length next to that first bullet point. Can be located at the tips of roots and shoots, in which case the roots get longer and the shoots get longer, or can be located inside the plant body, and I would write down diameter. It's how you get from a pencil-sized sapling to a tree that could be several feet in diameter. All right, so when we take a look at these meristems, though, they consist of two parts. Meristems can consist of initials, which are the actual cells that divide, and derivatives, which are the products of those cell division. So before I leave this slide behind, let me go ahead and take a quick look at the diagrams I've got for you to the right. That green thing, upper set of pictures on the left, is the diagrammatic tip of a shoot. And where you see the little red dot is where you would find a meristem. That's where all of the cell divisions are taking place. That's where all of these things called initials are located. And as they divide, they're going to create derivatives. And those derivatives are going to make up all of the other cells of the stem, all the other cells of the leaves that are starting to form. You could draw a little arrow and indicate that thing is growing upwards towards the top of the slide. So as the cell divisions are occurring, it's growing ever upwards. Now, to the immediate right of that picture, I see a root tip. A little bit more simple, but it's the exact same thing. Where the red dot is where the meristem is located. That's where all of the cell divisions are taking place. And all the cells produced, called derivatives, from the division of those initials are creating the cells that we see upwards in the root. So that root is growing downwards. It's growing ever downwards, generated by the cell divisions created in that apical meristem. So whether we're looking at the shoot on the left or the root on the right, these meristems are located at the tips. Now let's take a look at the illustration immediately below. I see two pictures side by side. They're basically the same plant. It's a stem in cross section. And so the stem on the left is younger than the stem on the right. Now what I want to notice is in this picture you will see what appears to be a really thin green line. It's in a ring, and there will be yellow tissue to the outside of that really thin green line, and there will be red tissue to the inside of that really thin green line. Now that little thin green line is a ring of cells that will be actively dividing. It is a meristem. But you will see that it is literally embedded inside of the stem. It's not at the tip. It's right smack dab in the interior of the stem. And so looking at that really thin green line, if you've got cell divisions going on, you're going to be producing new cells to the inside of the stem or cells to the outside of the stem. In any event, as those cell divisions are going on, that stem should increase in diameter. So if you take a look at the stem on the left, what's going on is that little thin green line is going to produce a lot of xylem, red tissue, to the inside of the stem. And it does that every single year. Every time you look at a tree trunk that's been cut across, you see growth rings. And those growth rings correspond to the amount of tree wood that is added every single year. You want to know how old the tree is? Count the little rings. So if I take a look at the illustration right, I notice that that particular stem shows me 2 layers of red tissue. That's a stem that's 2 years old. So that thin green line, that meristems, created one additional ring of growth above and beyond the original ring that was there. And you notice that the stem has increased in diameter. Go through the next year after that, next year after that, you're adding layer upon layer upon layer of the xylem tissue, and therefore the plant is going to increase in diameter. Now, the thing is meristems that are found inside of the plant body are commonly called lateral meristems. I think we're going to mention this on another slide, but I'm going to mention it right now. Apical meristems at the tip increase length. The shoot grows taller, the root grows deeper. But lateral meristems increase diameter. And so as the cell divisions are occurring in the lateral meristem, the plant gets greater in diameter because of all these new cells that are being created to the inside of the plant part. So let's take a look a little more at this basic idea here. So let's continue this discussion about where plant cells come from. We're also going to try to get into the issue of differentiation. Now you'll see the first bullet point here tells me that plant cells demonstrate what we're going to call totipotency. Totipotency means that a plant cell could conceivably become any cell in the entire plant. It's got total potential. That's what it means. You create a derivative from a cell division of initial. That derivative could be any differentiated cell in the plant. Could be a xylem cell, could be a phloem cell, could be a flower cell, could be any type of cell. And you'll see that in the diagram to the immediate right. Now that's a huge difference compared to animals, because in animals, for example, we'll take human beings. You've heard of stem cells, and you know that stem cells divide. Now, thing is, the stem cells are limited in terms of what they produce. So we're going to say that stem cells are pluripotent. That's Pluri. So it's what we would call pluripotency. What that means is it means a stem cell will and can produce a number of different types of derivatives, but it's limited. So for example, one stem cell might be able to give me 3 different types of blood cells, but won't be able to get me a muscle cell and won't be able to get me a bone cell. So there's a little bit of a limitation with animals, but the sky's the limit as far as plants go. Plant cells demonstrate totipotency. That means there's got to be factors that influence the differentiation of a plant cell into some differentiated mature cell type. I mean, if any cell can potentially become any cell in the plant, something's going to guide that development. So I got a bullet point that says here differentiation due to location, proximal cells, proximal tissues, the environment, hormones. In other words, if you've got a cell being produced at the tip of a plant, it's way above the ground, it's probably not going to become a root cell. As a matter of fact, if it's got a lot of other cells that are destined to become leaves or other leaf cells, it's probably going to become a leaf. If there are phloem cells around it, it might become a phloem cell. Okay, based on the environment, maybe how moist it is, how hot it is, other conditions of the environment might influence exactly what that cell's going to become. Let me give you another example. Let's suppose a tree falls in a forest. And when it falls, you might get the formation of adventitious roots. Remember, I mentioned adventitious roots. You can have some trees that actually create adventitious roots. If they're laying horizontally and they're in contact with the ground, you will get the novel formation of adventitious roots that will actually promote that tree's being able to stay alive. Because obviously when it fell over, its existing root system has been turned up and out of the ground. So that plant's going to struggle to survive, and by virtue of its position in its environment, we've got this unique differentiation of cells. And then finally, plants do produce hormones, and those hormones are commonly called plant growth substances, and they can actually influence the development of a derivative into some mature cell type. Now, what's really nice about this flow chart next to this set of bullet points here is it shows me the initials that are dividing, the derivatives that are produced, and the further back you go down from the stem tip, or the further back you come from the tip of the root, those derivatives are going to start differentiating. They're going to become mature cell types. And that's what contributes to the overall growth of the plant. So in your mind's eye picture, you're understanding the plant grows taller, the plant grows deeper, and if you're lucky enough, you got some meristems inside the plant that are causing it to increase in diameter as well. Now, I've been making the point that some plant growth results in an increase in length of plant parts, and some plant growth results in an increase in diameter. We've got some terminology to lay on this idea. Primary growth is growth that results in an increase in length. Primary growth is a feature of all plants, both woody plants and herbaceous plants. It is the ultimate origin of all cells because it's generated at apical meristems. And those apical meristems could be found at the tips of shoots or at the tips of roots. But the bottom line is any cell division going on at the tip of a shoot or in the tip of a root is going to ultimately be the source of all cells arising from that apical meristem. Doesn't matter whether it's a stem or a root itself. And so primary growth increases the length of the plant part. The roots grow longer, the shoots grow longer. The roots grow deeper, the shoots grow taller. So what about increase in diameter, Dr. Knauss? Well, increase in diameter is caused by secondary growth. Secondary growth is only found in some plants. So whereas primary growth is found in all plants and is the origin of all growth in a plant, secondary growth is found only in woody plants. It's not found in herbaceous plants. Now, the logic is, in order to become a woody plant, like a tree or like a shrub, you're going to need to generate a significant amount of growth internally. It's not enough to have the shoots grow taller. It's not enough to have the roots grow deeper. You need to get some increase in diameter in those shoots, some increase in diameter in those roots. And so this thing that we're talking about, secondary growth, is generated at those lateral meristems we mentioned not too long ago. Lateral meristems are embedded in the plant part. And lateral meristems we're going to see come in two different flavors. There's a type of lateral meristem called a vascular cambium, and there will be a type of lateral meristem that we're going to call a cork cambium. And the details of those are going to be found on our next slide. But right now we're understanding that secondary growth is a feature of only some plants, more specifically woody plants. And its job is to generate an increase in diameter or girth of the plant part. That's why shrubs and particularly trees have such a great diameter. Let's take a look at this lateral meristem called vascular cambium. The word cambium is almost synonymous with the word generator. It's not specifically the same, but vascular cambium is going to be a generator of vascular tissue. It is a meristem, and its job is going to be primarily to produce something called secondary xylem. It will also produce something called secondary phloem, but it will produce secondary xylem in large quantities, and it's that secondary xylem that is going to increase the diameter of that stem. Let's take a look at the picture at right. That is a cross-section through a tree called basswood, and that one that we're looking at is only 3 years old. You will see kind of a pithy center in the stem, and then you'll see 1, 2, 3 rings of xylem, and those 3 rings of xylem have those light blue arrows over them. And the light blue arrows are indicating the addition of xylem on a year-by-year basis. The ring of xylem right next to the pithy center is the oldest ring. The second ring is a little bit younger, and the third ring is even younger than that. And the reason why the third ring is younger is because that line where you find all of those light blue arrows coming from, that line is where the vascular cambium is. So vascular cambium is a lateral meristem. It is a bunch of meristematic or mitotic cells. It's a bunch of initials embedded in the plant stem. And when those initials divide, they are going to create secondary xylem. Now, the reason why we call it secondary xylem is it's being produced secondarily. If it was xylem being produced at the tip of a shoot or at the tip of a root, I guess we would call it primary xylem. So secondary xylem is being added secondarily. It's being added in an inwards fashion. And that secondary xylem, folks, I need you to know that is what we call wood in a tree trunk. If you look at a tree trunk or you look at a 2x4 or any piece of wood, that is technically secondary xylem. And I see the arrows indicating it's being added inwardly, and it's going to be added inwardly on an annual basis. And so those rings of xylem are annual rings. Now take a look at the close-up view, bottom right. An annual ring consists of two different types of wood depending on when the wood was created during the year. The first type of wood is called spring or early wood, and you see that the arrow is pointing to a section of a tree showing you that the cells are kind of big and large diameter, and that's what you would expect during the spring. Growth conditions are great, and so you're going to produce cells that are relatively large, and that's the characteristic of spring wood. Now, as spring moves into summer, growth conditions aren't so great. It gets really, really hot, and available water becomes few and far between. And then you get in the fall, and growth pretty much shuts down. So if you take a look at the second bullet point under annual rings there, it says summer or late wood, follow the arrow, shows you that the cells are significantly smaller and a lot more dense. So it's a combination of those two layers, spring or early wood and summer late wood, constitutes an annual ring. That's the tissue of secondary xylem laid down during a single growing season. And you can study trees and look at their rings and determine the actual conditions during any given year based on how thick the ring is and the characteristics of the tissue within that ring. All right, now this vascular cambium, not only is it going to be producing secondary xylem to the inside of the stem, but we got to admit it is going to be producing something called secondary phloem to the outside of the stem. You'll remember, it wasn't too long ago that I said that xylem, as a rule, is found towards the inside of the stem, and phloem, as a rule, is found towards the outside of the stem, and I said it would have implications a little bit later in the presentation. Well, that's where we're at. If you take a look at the inset picture where it says secondary phloem there, the outside of the stem is to the right, the inside of the stem is to the left. You will see at least two layers of secondary xylem in pink there, and then you'll see the secondary phloem being added to the outside. Now, where the pink and the orange intersect is where our vascular cambium is going to be. Now, to be honest with you, the amount of secondary phloem made is not anywhere near as much as the amount of secondary xylem made. So as the year goes by, and the next year and the next year after that, the far majority of increase in diameter is accomplished by the production of secondary xylem in those annual rings to the inside of the stem. The amount of secondary phloem produced is kind of negligible. All right, so there's only one other thing on here that I can think of, and that is secondary phloem also makes part of the bark of the tree. If you look at that cross-section through the basswood I've been talking about, where the little light blue arrows are, you'll see that dark ring outside of the xylem is primarily phloem. Then you'll see a really, really, really dark line around that, which is the actual periderm, or the outer layer of the bark. But now, if that was a tree, what constitutes bark for you and me is the real dark periderm layer around the very outside of the stem that I mentioned not too long ago. and a secondary phloem layer immediately inside of that. That's what you and I would refer to as bark. It's the bark on a pine tree, it's the bark on an oak tree, it's the bark on a maple tree. Here's another view of a stem in cross-section, obviously demonstrating secondary growth because I see a number of annual rings, and in this case I've got my pith at the far left. I've got 1, 2, 3, 4, 5, 6 different years of growth. And you notice some of those annual rings are pretty thick, and there's one that seems to be not thick at all, which probably indicates there was some basic problem during the course of the growing season, maybe a drought, or maybe in any event, we see where the vascular cambium is, and I see there is a really, really narrow line of phloem, or more specifically secondary phloem, that's just outside of the vascular cambium, but outside of that is this thick layer that they have bracketed labeled as bark. And that bark is a combination of the secondary phloem and what we're going to call the periderm outside of that. Now, the actual bark is primarily dead tissue, and that dead tissue is created by cell divisions in a second lateral meristem that we're going to call cork cambium. Now, remember, I told you the word cambium kind of means generator. So a vascular cambium generates vascular tissue, secondary xylem to the inside, secondary phloem to the outside. Now, a cork cambium is a cork generator. So what's cork, canals? Well, cork are dead cells. They're cells that are dead at maturity, and their job is, by virtue of their physical presence, to be a protective layer for the stem. See, the issue is when this tree was really, really young, the stem would have been surrounded by a layer of epidermis. That's all fine and good, but as the stem gets bigger, as it increases in diameter, that epidermis is going to split, and it's no longer going to provide the protection that you need. So what you're going to need is come up with some other way of being able to crank out protective tissue on the outside of the stem while at the same time keeping pace with the growth of the stem. See, one of the reasons when you take a look at a tree and you look at the bark, and that bark looks like it has fissures or cracks in it, it's because that bark has been fissuring and cracking as the tree gets larger and larger in diameter, but way down where that bark actually meets the wood, you're generating more and more and more of it, and it's staying consistent with the growth of the tree. So we're going to understand that bark is a combination of secondary phloem and the cork that's produced outside of it. And that cork consists of dead cells produced by this second type of lateral meristem called the cork cambium. So the results of secondary growth is a potentially massive plant that not only has capacity to transport large volumes of water and nutrients and minerals upwards, and significant amounts of food and sugars both downwards and upwards as needed, it also actually provides that plant with a structural ability to maintain its leaf position so it's able to maximize photosynthesis as well. And when we take a look at the organization of vascular tissue within the trunk itself, which is a stem, I see that secondary growth has given us a number of annual rings. So secondary xylem is added in an annual basis, incrementally, year by year by year by year. The older layers of secondary xylem, the older wood tissue, is deep inside of the trunk, whereas the younger wood is further outwards towards the periphery. And the reason is because that vascular cambium is located out towards the periphery of the stem as well. And then when you take a look at the tree, you see all this bark on the outside, which is this protective layer, which is constantly being regenerated not just through the action of the cork cambium because it's producing those dead cells, but also got to understand there is some production of secondary phloem as well by the vascular cambium.